So, good evening to one and all. As Rohit said, my name is Sarita Pandey. Currently I'm working as assistant professor in the New India University. It's in West Bengal. And today I am going to discuss something which is uh, just about the seed. It's a basic input of the agriculture. So before we do any kind of agriculture experiment, any kind of work in the field of agriculture, we have to take care of the seed with which we are dealing. So seed is the basic input and it is very much important for any kind of cultivation, not only in plant breeding, genetics, you go for agronomy, horticulture, any branch of agriculture, seed is the basic input and it holds a lot of importance to all of us. So here I'll be dealing with the many topics like what is seed, what is grain, and what are the different acts, why we are so much giving this importance to the seed quality, how to maintain that quality, and how it is going to help us to improve the production ultimately. So as we know, seed, if I ask what is seed, anybody will say that it is an embryo, a living organism, which is in the supporting of the food storage tissue. And it is also a basic input which is required for the any kind of experiment, especially in the genetics and plant breeding, because we have to improve the uh, plant in order to deal with the changing circumstances. Like nowadays, we are seeing that temperature is going so up and higher. So we have to have a variety which can give us a better production in such kind of environment. And if you go through the different parts of the India, all the part of the India holds different kind of conditions. So we have to develop a variety for any crop that can survive in those adverse conditions and can give us a good yield. So if you go by the biological definition of the seed, it is nothing but a ripened ovule or a fertilized matured ovule containing embryo which has developed under fertilization. But if you think from the seed technology point of view, what is a seed? We will say any part of the organ which has the capability to regenerate into a new plant. Like if we say that seed, then any grain can be a seed, right? But when I say about, so just think about a ginger, then the rhizome is the seed. Think about the plant which propagates through the leaf cutting or the stem cutting. So in that case, the seed is that particular part of the plant. So seed definition will differ. If you ask a biologist, if you ask a seed technologist, so the seed definition will change as per or his expertise. So initially, we used to call any grain or any seed which can give rise to another plant is a seed. But in 1966, a Seed Act was implemented by the government of India, where the definition was given what is actually a seed. Seed of a food crop, including edible oil, fruits, and vegetables. Everything comes under this definition of a seed. Even initially, cotton seeds and jute seeds were not included in the seed definition. They were also included in the Seed Act in 1966. Any seedling, tuber, bulb, rhizome, roots, cutting, all type of grafts and other vegetatively propagated material for food crop or a fodder, those were all included under the definition of the seed in the Seed Act 1966. So before we go into the detail, how will we differentiate between a seed and a grain? If I have just given this picture, in this handful of rice grain, can you tell me which one is the seed and which one is the grain? What will make the difference? Which particular unit will be regarded as a seed and which one we will be saying that no, this is not a seed, this is a grain? So that definition has to be defined. If it is a seed, it has to be viable. And if it is not viable, we can consume it anyways, right? So that can be regarded as a grain. So when we are differentiating two particular grains of a rice, one as a seed and the other as a grain, we have to have some definitions in the place to define them into two categories. If it is a seed, it has to be viable. It should have a maximum genetic and physical purity. It should have, minimum seed certification standards, and it should be treated with the pesticides or fungicide because we are going to store them for the next season. Respiration rate and other physiological, biological processes should be kept very low because we need to keep them viable for the next season, and should compulsory be certified or truthfully labeled. So if these categories are being fulfilled by that particular unit, we will be regarding it as a seed. Otherwise, if it is not viable, if there is a not so high maximum genetic or physical purity, there is no need of any kind of certification. There is no certain things that we have to keep the respiration or biological processes under the check. So in that case, we can call it a grain, because the only purpose it is going to serve is for our consumption purpose. We are going to consume it. So there will be no treatment by any fungicide or insecticide. So this is a very simple definition, but most of the time I'm just keeping our undergrad students for this. Like, we generally forget what is seed and what is grain. So seed is viable, which is going to give rise to another plant, and seed and grain is something which we are going to consume. Now I was emphasizing on the word seed quality. It has to be maximum physical purity, genetic purity. So what is seed quality? Seed quality is the possession of seed with required genetic and physical purity, and also with the physiological purity. So generally there are three things which we have to take care of. First is the physical quality. Physical means what? Whatever we see. Like, the seed lot has to be clean. It has to be clean from what? In what sense we are saying that it has to be clean? A seed will normally be very clean, right? But there should not be any other seed. There should not be any debris, inert matter, disease seed, insect damaged seeds. So now the term comes, what is inert matter? I think most of you know what is inert matter, but just to clarify, inert matter is anything which is not seed, like any leaf, part of a leaf, part of a stem, soil, sand, some stone, plastic particles, anything other than the seed will come under the category inert matter. So we'll have to keep them under the check, because when you are doing a crop production is entirely different thing, and seed production and seed technology is entirely different thing. In seed production, we deal with the quality. In crop production, we deal with the quantity. Our quantity has to be higher in case of crop production, but we do not care about much about this physical purity or genetic purity. But in the case of seed technology, where we are dealing with the seed production, quality seed production, so in that case, we have to keep in mind that whatever we produce, the quality has to be of a good standard. Otherwise, during the certification process, our seed will get rejected. So this, most of the time, this inert matter and everything has to be below 2%. Like, if we have a packet of 100 kg seed, it will be less than 2%. So very minimum fraction of something can be allowed in a packet. Otherwise, it will not get certified during the certification process. Second thing is genetic purity. Physical purity is something which appears. Like, we can simply say ki in this particular seed lot, I can see leaf particle, I can see plastics, I can see I can see stem, I can see flower birds, etc. Seed, sand, anything. But what about the genetic purity? Can we examine the genetic purity just by looking at it? Is there any way to test the genetic purity? There are some ways through which we can test the genetic purity. Genetic purity means inherent characters that that particular plant will have, and it will transmit that from one generation to the another generation, and it will keep it true to the type. If I'm talking about IR36, it has to be IR36. There should not be any mixture of other crops, other varieties looking like IR36. So this is called genetic purity. So how we are going to test is the genetic purity? There are many methods. So two very common method is, like, if I have a good lab, I have a PCR and I can do the DNA fingerprinting, then I'll go for the genetic purity by using the molecular markers. Nowadays, there are very good markers available. The very simple one is SSR. SNPs are also there. So we can apply them and we can test the genetic purity. But if we do not have such kind of environment lab right now, then we can go for grow out test. GOT in the short form we call it. We can perform that test in the field where we can grow the true type of the genetically pure line, and the mixture which was given to us for the testing purpose, we can compare them, and then there is a whole standard protocol for this, what we have to check, how we have to check. For each different crop, this protocol is different. So that is not included in this particular session. We will be discussing it later on in some other session. So we can do the genetic purity test. So these are all the work of seed testing lab. There are many seed testing lab all across the India, and farmers generally give their seeds there, researchers also, and they perform this test. So genetic pure is the second one. Third is the physiological quality. What is physiological quality? It is the actual expression of seed in further generation or multiplication. Like I have a seed which is physical, which has a good physical purity, like very clean, no debris, nothing, no disease, everything is good. Also, it is from the same genetic purity it is having, just like a mother stock. From the stock from where I have taken the working sample, it is same as the mother stock. So genetic purity is also 100%. But what about the physiological quality? If the seed do not germinate, then there is no use of this physical quality and genetic purity, right? So we have to test the physiological quality as well. Physiological quality, can I have the questions at the end? So that would be better. So we will be having three types of physiological quality that we need to check. The first one is called germination. Like whether viability. The first one is viability. Viability means it's like whether the seed is dead or alive. Means the liveliness of a seed is called the viability. If it is viable, then it will germinate. So viability can be tested through the germination test. One is the simple germination test, and the other one, the chemical test, is called tetrazolium test. Once the seed is viable, then we will see whether it has this germination percentage. What is the germination percentage? Because in seed production, for each and every crop, we have a minimum standard of the seed germination percentage. Like if it is rice, it has to be 95%. If it is a cotton, it has to be this. So we have to fulfill that particular one in order to pass the certification process. The third one is seed vigor. What is the vigor? It is the rate of emergence of seedling, how fast the seedling is growing out of the seed. That is called the vigor test. So three physiological quality we have to check: viability, germination, and the vigor. And based on this, we will be giving the, how good quality is of our seed. So why we are so much bothered about the quality seed? And what characteristic a good quality seed should have? Like based on my previous slides, it should have a high genetic purity, high pure seed percentage. It means physical purity, good germination, vigor, higher field establishment, free from any kind of pest and disease. All should be very uniform, and high shelf life, optimum moisture content. Optimum moisture content. Moisture content is very important for the quality of the seed. Why? Because during the storage time, if the moisture content is not on the lower side, then there will be attack of this pest and disease, pathogens, and the physiological quality will deteriorate. Therefore, we have to keep the moisture content very low. But there is also a fixed percentage of this moisture content for different type of seeds, for cereals, for pulses, for vegetable crops, for oil seeds. So in seed production, we have to go by that protocol. Whatever crop you are choosing, for that particular crop, we have to maintain that particular things in mind. So if we have the good quality seed, what is the significance, right? So it will ensure the genetic and physical purity. We will have the desired plant population. It can withstand the adverse conditions also. It can grow more vigorously. It will ensure the uniform growth and maturity, because if there will be no uniform growth and maturity, then there will be a problem in assessing the different stages of the growth, which is very important for the seed production. And then there will be, because during the entire seed production, if I am a farmer or anybody else who is growing a particular seed for seed certification process, during the entire crop life cycle, there will be a visit of seed inspectors from the organization, seed testing organizations, right? And they will be coming and visiting our plot. So if they will not find it uniform and as per the protocol, then there is a chances that our particular plot will be rejected. So we have to keep that in mind. Second, now is the seed and its multiplication. So what is seed multiplication ratio? Seed multiplication ratio is very simple. It is the ratio of seed yield per seed generation. That is, many seeds are produced from us. How many seeds? Like in case of rice, it is 1:80. If you plant one seed and it is the average one, it is not the exactly, you will not find that you will plant one seed and it will give you 80 seed. It is the average of different varieties and different type of situations, circumstances, like conditions of the soil and where you are growing. And what is seed replacement rate? It is called SRR. Seed replacement rate is the quantity of quality seed that have replaced the actual seed requirement of the location that are normally produced by the farmer using their own seed. Right? So SSR, we generally calculate X by Y into 100. X is the quantity of actual quality seed sown in that particular area, and the Y is the quantity of quality seed required for the entire production area. So these two ratios are very important for the students who are, studying seed technology or plant breeding, or will be appearing for net or any kind of, because they give this multiplication seed replacement ratio for many of the crops. So suppose a farmer or anybody who has bought a packet of certified seed and he or she is growing in his particular environment. But after few years, you will find that there is a variation in that particular seed lot. It means the quality of that particular packet of seed lot has deteriorated. So how come that is possible? Because if it is a certified seed, it has to pass through all the criteria which is given there, like minimum germination, minimum percentage of inert matter, this and that. So there are a lot of too many. I will come to that. So if we have purchased a particular seed packet, certified seed packet, and we are growing, then how come it is that that particular seed is getting deteriorated in quality wise? So there are 7 different conditions, or you can say factors, that helps to deteriorate the quality of the seed. The first is called development variation. Second is mechanical mixture. The third is the mutation. Fourth is natural crossing, minor genetic variation, selected influence of pests and diseases. And the last one is the techniques of the breeder. So I'll discuss each one of you. The first is the development variation. What is development variation? Suppose I have purchased a particular same packet of the seed and you have also purchased the same packet of seed, and we are growing that particular seed packet in a different environment where it is not supposed to be grown, or where it will not give you the desired yield or the quality which we are looking for. So in that situation, what will happen? Obviously, it is a good quality seed, so it will germinate, it will give you a good plant stand, but there will be some variation, right? So that variation, that minor variations are called developmental variation, which is generally because of growing them under the different conditions like soil fertility, climate, photo period, different elevations for several consecutive generations will give rise to such kind of development variations. So therefore it is advisable that for any part, like, when the variety is being released, it is said that this is for the northern part of the India, or this is for this particular state. So we should generally follow that thing in order to keep the quality under the check, right? And there will be no variation or less variation. The second one is called the mechanical mixture. And this is the one of the most important thing. What is mechanical mixture? Mechanical mixture, like when we are growing something, suppose I have purchased a very good quality of seed and I am growing in a particular plot. But when I'm walking through the different plots, I can carry some seeds, like in our dresses or in shoes or the instrument or things which I'm carrying with me. All those things can carry some other seed or other variety. And if it is of the same crop, it will be very difficult to understand just by looking at the plants. Like this is from the IR36 and that particular seed is from IR64, right? It is not possible, in at least in the very early stage. Once it goes to the flowering stage or in the reproductive stage, you can have an idea like this is not the exact one, or this is not behaving like the IR36. Suppose if IR36 the main crop I'm dealing with, so there you can differentiate, but in the very beginning you cannot differentiate. So such kind of mechanical mixture has to be, taken care of to avoid quality deterioration. The third one is the mutation. Mutation is not a very serious issue because, in natural mutation chances are what, 10 to the power minus 4 to 10 to the power minus 6. So it's a very, very low. And if there is a mutation and if we can see it in the field, we can just remove it during the roking process. Natural crossing, this is another major factor that leads to this such kind of deterioration of the quality. What is natural crossing? The crossing between the two different type of the same crop, right? So like suppose I'm growing IR36 and IR36 is my main crop, and just nearby, near to the my plot, there is IR64. And because of wind and other thing, the natural crossing is going on. So what will happen, without my notice, there will be some seeds which are the hybrid one, natural one. And it will be very difficult for me to differentiate. But during the genetic testing, genetic purity testing, we can simply see, or if there is a morphological variations, then it will be very easier to detect them during the time of physical purity test. So we should be how to like what measures we can take in order to keep this natural crossing in under control. We can give the isolation distance. What is isolation distance? Isolation distance is a fixed distance that has to be given between the two plots. Like rice, for example, it is a self-pollinated crop. So we have to give 3 meters all around the plot as an isolation distance in order to check this natural crossing under the control. So this is the major one. The next one is the minor genetic variation. Minor genetic variation can occur even in varieties appearing phenotypically uniform and homogeneous. Like suppose I have one breeder has released a variety and he or she has released it little earlier. So it was appearing phenotypically uniform and homogeneous, but it was not 100% homogeneous. Still some variations were going on. And the moment we plant it, they are showing some variations. So that particular lot will give rise to minor genetic variations. The sixth one is the selected influence of pest and diseases, like new crop varieties, which are like susceptible to the new races of pest and diseases. They may show some kind of variation because of that infestations, right? Or infection. So these type of influence of the pest and diseases may lead to some minor genetic variations. And the last one is the technique of the plant breeder. At the end, we all are human being and there is a chances of, making mistakes. So sometimes the technique which is being implemented by the plant breeder may also lead to some kind of genetic variations. So these are the major reason or factors which leads to the deterioration of the seed quality. So if we are keeping them or keeping a mind like eyes on them, there should not be any kind of natural crossing. For that, we have to give a isolation distance. We should carry the things properly to the field where we are growing for the seed production, because this is a small field, and that field has to be taken care of very, very strictly. Otherwise, what will happen at the end of the entire process, it will be liable for the rejection. So better to keep these things under the check. So now the question comes, how we are going to keep the genetic purity, how we are going to maintain this genetic purity. Simply use of approved seed, like you need to buy always the certified seed from the authentic source, not from the market. You have to see the tag. We'll come to the tag, what is tag and how to take care of all those things. Inspection and approval of field prior to planting. Wherever we are planting, we just cannot choose a plot. Like in the case we do for crop production, in the seed production, the things are little different. Like suppose I'm going to plant my crop in particular, I have to look for the history. In the last season, what crop was planted there, whether it was from the same type of crop. If same type of crop, what was the variety, whether the same variety or the different variety, whether it was for the crop production or for the seed production, whether certified seeds were used or the normal we purchased it from the market and we planted it. So all these things has to be taken care of in order to avoid what? Because the previous season seeds will germinate, and those are the volunteer crops. So we have, and they can also lead to this natural crossing. So we have to keep the natural crossing under check, and we have to keep them in such a way that there should not be so much contamination or admixtures in the our stock, because physical purity parameters are given there. 98% physical purity we have to maintain for most of the crops. So it is a very higher percentage, and we have to avoid any kind of contamination. Otherwise, our field will get rejected. We have to get our field inspected on time to time, and especially on the critical stages. Second, sampling and sealing of the cleaned lots. Whatever lot we are taking from the field, we have to sample it, we have to clean it properly, and we have to maintain that stock properly. So this is how we can maintain the genetic purity of the seed. So simply, genetic purity, like I told you or earlier, I have already explained all these things, like isolation distance we have to give. Roguing, what is roguing? Roguing is a removal of off type from the field. Off type, what is off type? Like suppose rice is being grown in the plot. My crop is IR 36. I know that IR 36 will be height of 110 centimeter, for example. But there are some plants in the same stage where they are showing me the height of 150 centimeters or 30 centimeters. So I don't know. They might be the same variety, can be a different variety, or something else. So we will not go into any kind of doubt, and we will simply pull them out and throw them out of the plot. This process is called roguing. They may be from the same lot. They might be the IR 36 because of certain other things or something different happened to them, and they are showing such kind of variation in the height. I'm just giving the height example. There are few more things like flowering, branching, anything can happen. So we have to keep our eyes open, and if there is a variation which is morphologically we can identify, we have to identify such plants and remove them, so that we can reduce this off type percentage. Because off type percentage, for this off type percentage also there is a thing like 0.1% only we can allow for certified seeds. So this is how we have to maintain everything in order to get our lot passed through this certification process. And this generation system, I will explain this generation system. Grow out test I have already covered. So seed production, so now you might have understood what is seed production. Seed production is nothing. It is a crop production, but in a systemized way. There is a way of doing everything from selection of the plot to the harvesting and post-harvesting things, what we need to do and how much, what is allowed, when has to be done, how it has to be done. Everything is defined. And at every stage, there is an inspection and there is a report. So it is the same crop production, but in a systemized way. And the target is to have a good quality seed instead of a good quantity seed. Obviously, if the quality is better, the quantity will be good. We will be getting a good price for whatever we are producing. But here the focus is quality, not the quantity. And if the quality is good, obviously higher income will be there and higher quality seed for the next sowing. If the quality is good, you will need a less quantity for next season of the sowing. So it is a time to be very much clear about what is seed production and what is crop production. So seed production is the basic, thing where we are going to have a very, very good quality seed. At the end, crop production, we are more into having a good yield. Here you have to seed product in if you are doing seed production, you have to buy the seeds from the authentic source. You have to carefully choose the plot where you are going to plant the seeds. You have to give the proper isolation distance. You have to maintain this, all kind of technical things like how much area, what is the seed rate, what should be the row to row, plant to plant, and exactly you have to maintain. You cannot make any modifications until it is allowed. Roguing is compulsory in case of seed production. We do not go for roguing in case of crop production. We are not so stringent about the quality parameters in case of crop production, but we have to maintain that in case of seed production. So whatever things we are talking about in a seed production, it is not like it is not an optional, it is mandatory and you have to do. In crop production, those things are not mandatory because we are not going to get, or our target is not getting the higher quality seed, but we have to get the good quantity of the seed. Now, in the seed production also, there are two different type of seed production. First is the varietal seed production and the second one is hybrid. Variety is like any variety, we have to multiply. And hybrid means we have to cross two parents in order to get the this hybrid seed production. So in case of varietal, it is little simpler because it is a single parent multiplication. Isolation requirement is also less. Production is just by open pollination. And the seeds can be used for 3, 4, or 5 generations. Production techniques is also very simple and uniform. Production care is on the lower side. And here the yield is little lower than the hybrid one, and profit is also low because in hybrid the production will be higher, the profit will also be higher. But in case of hybrid seed, we need 2 or more parent. Isolation distance requirement is also very high. Production has to be maintained by pollinations, and seeds has to be changed every time, and technical expertise is also higher in case of higher seed production. So this is not very important. In 2002, the national seed policy was implemented, and during that, some trust areas were identified, and we are still working on it. Like we have to be, we need to do varietal development. We have to do more seed production. We have to increase the seed replacement rate. Like we have to encourage the farmers to use good quality seed and the new variety, which are superior than the earlier one. And we have to use the effective seed production program, popularization of the new varieties, availability of newly developed varieties to the farmers, right? So these are the things that we have to do in seed production, and government is doing its best to, popularize the new varieties and making them aware about the new technologies, making the seeds available to them to different channels where they can go and they can buy the authentic seeds so that the losses will be less. So these are the areas. Now, what are the classes of the seeds? From the very beginning of my slide, I'm talking about certified seeds, and certified seeds. So is there only certified seeds available, or there are many classes, other classes of the seeds? So generally there are 4 classes of the seeds. Here they have, if you go by the definition, theoretical, so there will be 4 classes of the seeds. First is called the nucleus seed. The second is called the breeder seed. The third one is called the foundation seed, and the fourth one is the certified seed. So nucleus seed is what? Nucleus seed is the seed that, suppose I'm crossing 2 parents and I'm getting the F1. The very beginning, the very first seed that a breeder will develop is the nucleus seed, and it is very less in quality, and there will be no multiplication by the national or state seed corporations. The second one is the breeder seed. Breeder seed is produced from the nucleus seed under the supervision of the breeder or the research institute where the particular seed was developed, and it has the 100% of the genetic purity. And there are different colors of tag which we particularly use for one particular class of the seed. For breeder seed, it is a golden yellow color tag. So this type of tag, if we find on certain packet, we should understand that this is the breeder seed. And on the any tag, there will be certain information which is written, like crop, which crop is this, which variety is this, which class of the seed, lot is what, date of test. We do all kind of physical purity, genetic purity, physiological purity, and there are other tests also. So on which date it was done, what is the inert matter percentage, because that matters a lot. What is the germination percentage? What is the genetic purity? So all these things has to be written on that particular lot, and particular seed tag has to be given. Like this is a breeder seed, so yellow, golden yellow color seed has to be given. The third one is called the foundation seed. Foundation seed is the progeny of breeder seed, and it is produced by the State Farm Corporation or the National Seed Corporation. And same information will be given there. And here the genetic purity is 99.5%, and white color tag will be given. The fourth one is the certified. Certified seed is a progeny of foundation seed, and its production is supervised and approved by the certification agency. Here the color is azure blue, right? And here the genetic purity will be 99%. And it is normally produced by the state and national seed corporation or private seed companies or farms or any cooperative. If a progressive farmer is there, he can also produce the certified seed. So these are the different tags, like, which are available in the market. Nowadays, this registered seed is not included in this generation system. Generally, it goes from breeders to foundation to certified, and there are different subclasses of the foundation and certified seed. And there is one more class called TL seed, which is truthfully labeled seed. It is same as the certified seed, but little different. I'll come to that. So certified seed, here the certification is volunteer, and truthfully level seed is compulsory for all the notified varieties. Certified seed is applicable only for the notified, while truthfully level seed is applicable for both notified and released. Right? So they are quality wise, they are same. Certification seeds, they have the certification, and in truthfully certification is not required. Okay, so only difference is that generation system. Actually, we are quite 7:40. It's already 7:45, so I'm wrapping it earlier. Generation system of seed is what? Generation system of seed multiplication is the production of a particular class of seed from this specific class up to a certified scale. Like suppose I need to produce a breeder seed, then I'll have to take nuclear seed and produce the breeder seed. So it depends, like how many classes, what generation system I'm going to opt, that will depend on three factors: the rate of genetic deterioration, seed multiplication ratio, and total demand for that particular seed. So keeping these three things in the mind, we will have to opt for a generation system. So there are three generation systems. First is called three generation system. Then simple one. From breeder seed, I'll go to the foundation seed, and from foundation seed, I'll go to the certified seed. In four generation system, we'll go from breeder seed to the foundation one, then to the foundation two, and then to the certified seed. In the third one, breeder seed to foundation one, two, and then certified one and certified two. So these are the different generation system, and this we need to follow as per the demand and seed multiplication ratio and the rate of the genetic deterioration. So keeping these three things in the mind, we will go for opting this generation system. This particular table is, flow chart is very important. Like first is the nucleus seed. Nucleus seed does not have any specific tag, and it is maintained by the breeder who is developing. Second is the breeder, which has a golden yellow tag. It is maintained by the organization or research institute, agriculture university, and breeder himself. The next one is foundation 1 and 2, where state seed certification agency, field inspection and testing, etc. is needed. White color tag is provided. The next one is the certified seed, where other blue color seed is provided. And the last one is the truthfully level seed, where the farmer can grow, and truthfully level seeds are very much popular, and we can get it very easily. So this is all for this particular session. Apart from this, in seed testing, we normally do many things, like we study about the seed processing also. Once you harvest the seed, there should not be any mixture. We have to grade them. So what is grading system? Why grading is so important? What is the different storage per thing, like short term, medium term, or long term storage? Why moisture content is very important? How to keep this moisture content under the check? And then the seed treatment. What are the different seed treatments? Seed treatment is not only for the, fungicide or insecticide. It is also, there are many special kind of seed treatments for germination, viability, biofortification, etc. So we also deal with those things. Those things will also come under the seed production. So this is a very brief of what seed technology is all about, why the seeds are so important, why maintaining the quality of seed is so important, why we should follow the classes very firmly, what are the generation system, why this generation system has been developed, how it is going to help us. And apart from this, we also need to study here, or we also need to know about the different seed acts and seed policy which are implemented time to time, and what are those policies, what are the different things, or characteristic of those particular policies, what is the important points those are given there, how many national and state seed corporations offices are there, seed testing offices are, where are the different seed testing offices, what are the roles of these seed testing offices, and who are the seed inspectors, what are their role, what are the different clause of the seed acts. So all these things are part of the seed technology. So this is a brief one, a very small lecture on the seed technology and importance of the seed. I think we can have the questions now if anybody has.